Hello, this is Frank from Intel Minds, and today we're going to be talking about 1.3 membrane structure from the IBDP program. The main idea is that the structure of biological membranes makes them fluid and dynamic. First off, we want to start off with the fluid mosaic model. This was proposed by Singer and Nicholson in 1972 and is currently accepted as the best model for the phospholipid bilayers, also known as fluid, and the proteins, which are known as mosaic, hence the name. So the structure of phospholipids consists of a hydrophilic head, which is polar, and two nonpolar tails, which are hydrophobic, shown here in yellow. So the polar head is made of a glycerol and phosphate molecule, shown in the picture on the right, and the two nonpolar tails are mainly consisting of a hydrocarbon chain. So containing both hydrophilic and hydrophobic regions means that phospholipids are amphipathic. The properties of the phospholipid bilayer. So the phospholipid bilayer is held together by these weak hydrophobic bonds, and these are consisting of the tails, which are facing each other in the picture below, and they create these weak hydrophobic associations with each other. This allows the layers to restrict substances from entering the passage freely, although it also allows the individual phospholipids to move freely and fluidly throughout the membrane. This allows for membrane fluidity and flexibility. Hence, this is why we can see that endocytosis and exocytosis can occur spontaneously within the phospholipid bilayer. Next up, we have membrane proteins. So there are different types of membrane proteins that exist within the phospholipid bilayer, and they're important in regulating the activity within the phospholipid bilayer. So first off, we have integral proteins, which are permanently attached to the membrane and typically span across the bilayer. Integral proteins are usually seen over here, and they're partially inside and partially outside of the phospholipid bilayer, usually making contact with the hydrophilic head and sometimes the hydrophobic tail. Next off, we have peripheral proteins, which are temporarily attached by non-covalent interactions and associate with only the surface of the membrane. Hence, it would only be touching the hydrophilic head and not the hydrophobic tail. Nonpolar amino acids also associate with the lipid bilayer, and polar amino acids are located internally, where there are aqueous solutions. Next up, we have cholesterol. So cholesterol is a steroid found in animal cells and is very important. So first off, it maintains the integrity and mechanical stability inside the cell. It is also an amphipathic molecule as it contains both hydrophilic and hydrophobic regions. The main use of a cholesterol is that it regulates membrane fluidity and also prevents the crystallization. It contains of a hydroxyl group, or an OH group, which is hydrophilic, and this aligns towards the phosphate heads, and also contains a steroid ring and hydrocarbon tail, which are hydrophobic, and this associates with the phospholipid tails. Finally, we're going to be talking about the Davison-Danieli model, which was proposed as a central phospholipid bilayer surrounded by two layers of protein. This existed before the fluid mosaic model and was known as the lipoprotein sandwich. It basically assumed that the ratio of the lipid to membrane surface was 2:1. In other words, the section here shown in blue was double the section shown here in yellow. However, this seemed problematic as this was incorrect. First of all, it assumed that all membranes had a uniform thickness and a constant lipid-protein ratio, which is not true when we see the micrographs here today. It also assumed that all membranes had symmetrical internal and external surfaces, which are also not true due to the fact that we can see integral and peripheral proteins. It also did not account for the permeability of certain substances, and we also believed that the membrane was immobile. Thank you for listening to our video from Intel Minds. Like the video and subscribe if you found it useful.